Hello, and welcome to the Boffin Buzz, the podcast for anyone fascinated by farming's future. Hello again, and welcome to the Boffin Buzz, and part two of our interview with Kerry McDonald-Howard from UK AgriTech Centre. Hello, Kerry.
Hiya, Tom.
Great. Well, now before we go into the discussion, we are having a really great discussion in the previous episode about nematodes and all things to do with slug control. You know, how we can use nematodes for slug control, and we're going to be looking in that in more detail. But first, I just want to remind those people who are collecting CPD points that CPD points are now available for the Boffin Buzz. We gave the first part of the code that you'll need in the first episode. If you haven't heard the first episode, well, I think you better go away and listen to it now. But do come back and listen to the second episode that we're about to go into now. And at the end of this episode, we will give you the second part of that code that you will need to claim your CPD point. So anyway, as I say, we were talking, Kerry, in the previous episode about your work with nematodes and how they can be used for slug control. Now, Kerry, for those who haven't listened to episode one, you're an expert in slugs and pests, and you've got a particular passion for sustainable control methods. And you've got a degree in zoology from Bangor University and a PhD of improving the efficacy of the parasitic nematode of slugs as a biological control from Liverpool John Moores. And you describe yourself as a malacologist, nematologist, parasitologist, and entomologist. Oh, I got all of those out. Anyway.
That just all sounds incredibly insane. Especially the first part, expert on slugs.
which is expert on slugs and nematodes, I guess would be, well, that's certainly in terms of what we're talking about today.
So I'm learning new things every day about those.
Yeah. Now let's talk a bit more detail about slimers. So this is strategies leading to improved management and enhanced resilience against slugs. It's a £2.6 million DEFRA-funded project, and we've got, I think it's 7 industry partners, all told, including the UK AgriTech Centre, and Kerry works for UK ATC. Now, regular listeners will be familiar with this project. Basically, we are looking for different ways of controlling slugs. There is only one form of slug control at the moment, which is ferric phosphate pellets, which is and just one active ingredient as well. And we rely on that completely. It costs the industry about £43.5 million a year to control these pests. It is arable farming's biggest pest. And we're looking at 2 different areas. First of all, patch location of slugs, and we'll talk about that in a bit. And also using autonomous control of slugs. And this is where, Kerry, your expertise really comes in here. So we heard about your areas of expertise in the previous episode of Boffin Buzz. Could you just sort of talk specifically about your role in slimers and what you've been working on?
Okay. Well, I've come on board as a research associate, and I'm based at the UK ATC's phenotyping lab that they have based at Rothamsted, where I'm right now sitting, at Rothamsted Research Centre at Harpenden. So we have this lab here, and what we do is we run a load of different experiments here. In fact, we're running a number of them at the moment. So what we're working at the moment in the lab here itself. Should I go into a bit of work I'm doing at the moment in the lab?
Yes, no, please do. I mean, one thing that's interesting, for those people who don't know the UK AgriTech Centre, you've got a number of resources, I think, around the country that you use for research. And it's those facilities that you're using at Rothamsted, is that right?
Yeah, so we have got a number of different facilities, all the way from the bottom of UK to the top into Ireland, and we've got a number of different facilities and capabilities. So the phenotyping lab is why I'm based here, is because we're doing a lot of multispectral work, building AI modeling to train sets that we are able to distinguish slugs from different backgrounds and distinguish slug damage and different things like that. So that is the fun of actually being based at this lab, I've got a lot of fun
Yeah.
equipment that I can work with,
Well,
and I've got
just hold on a bit, because you
okay.
said multispectral equipment. So, are we talking about, is this camera equipment specifically we're talking about here?
Yes, it's camera equipment, but it's looking through different light spectrums. So it's not just
Right.
RGB camera; it's looking all through the different things, and it uses different light. It looks through the light spectrums, and it builds an image. And what we can actually see is the different plant responses
Right.
to. So if we've damaged the plant with a slug or damaged it with some mechanical device or not damaged it all. So we're
Right.
building modeling sets that we can then have an AI trained set, an AI system that can say, this is slug damage. And we're also
Ah,
building modeling sets where we're training, looking at things through different spectrums. So we've got rocks and we've got stubble, we've got wheat, we've got different plant materials and things, and then we've got a slug. And we want an AI to be able to say, this is slug, that is a rock.
right.
So
So
that's
now,
the work
and
we're doing in the lab, but it's very basic
yeah,
before
so
we get
just to
out into the
clarify
field.
here, what we're talking about is taking a camera out into the field, mounting it
Yes,
on a spray boom or whatever, and then
but
that camera can detect the slug. And
mm,
I guess the reason we're using multispectral imaging, this it's looking at all the spectrums of light,
yeah,
is so that we
and then we
can
can narrow it
define
down.
a slug from a stone, or
Yes,
from a
so
piece
we
of
can
trash
narrow
or
it down,
something.
and so we've got a sort of we're getting ahead and building the set so that we
Yeah.
can then actually get out into the field. Because what I think I spoke about Slugbot previously. So they were doing a
Yes.
similar thing, and they did a lot of lab work. But what we need now is more variation. So this is why we're going to start with working with Buff and Farmer Network, or we've
Yes.
started already. And I visited quite a few farms and started doing a few images and picking up what type of land, the layout of the land as such, what type of backgrounds we're looking at, whether there's rocks, whether there's no rocks, whether it's red soil, and all different soil types and things. And then we're going to have our set that we've built in the lab. We've ascertained what we're actually looking for, and
Right.
then we're going to have units that we're doing for data collection, where we're going to go out into the farms themselves.
I see.
That's our next steps in the next year, and we're going to actually collecting that data, building a strong set, so that when we've got the autonomous system, we can geolocate the slugs, and then we can actually monitor where the slugs are and precisely apply the nematodes. So that's the overall. We're playing in the lab doing the baseline stuff. We're going in the field getting the big training sets, and then
Right.
we're applying that to an actual autonomous system that we can go out, geolocate, find the slugs, and apply the actual control to the
Yeah.
slug directly.
And we're talking about nematodes here. Now,
Yes.
we were talking about that in part one. So, nematodes are tiny microscopic organisms that live naturally in the soil. We can apply them to the actual slug themselves. And we
Yeah.
talked in the last episode about how effective they are at control, but prohibitively expensive to apply willy nilly over your field.
Yeah.
So, this is why we're talking about real precision here.
And
And
if
in
we get
order
it
to,
right, much cheaper than chemical controls, simply
yeah,
because it works a lot better. They don't wash away, and it kills and reproduces.
yeah, absolutely. Really, really good method if you
Yeah.
can get it in the right place and the right conditions
Yeah.
and so on, which is
So,
what
so,
this project is all about, I guess, in
so
finding the
yeah.
right place and the right conditions. But I just want to rewind a little bit back
Mm,
to, and you mentioned the Slugbot project, and this was one of the reasons that we brought Simers together. That
mm,
we were aware of the previous work done by Dr. Jenna Ross, OBE,
yeah.
into with Slugbot and working with looking to detect autonomously using autonomous robots to
Yeah.
detect slugs and then apply. And I think they got, sorry, apply nematodes, that is,
Yeah.
and then and I think they got to a proof of concept
Yeah.
with that project, that you can actually
It
spot
proved the
slugs.
potential of it, and they
Yeah,
showed it. They could spot the slugs, and
yeah,
they could actually. They already developed spraying systems and that we're working with. So we've got a more precise spray. So we, I think we got it down to something about like under 30 centimeters area
right,
of precision spraying in that. But we are working now with the idea that we need more variability in the background of those
sure,
pictures.
yeah,
So there isn't anything that's going to trip up the AI, so that we've got the system out there that, because I think you all know that the farms are very different, especially with my trips being out recently across the UK. That was quite some different farmland from one farm to another. So we need that different data set built.
Yeah, no,
That's our next step.
absolutely. Just sort of moving on to the nematodes. Let's
Yeah.
talk about the nematodes themselves and why are we using nematodes with this autonomous system then?
Well, it's a brilliant synergistic approach. So you've got the nematodes, who are
Yeah,
a perfect control of the slugs, and you've got a autonomous system that can work day or night, but it can work in the environments that a person probably won't want to go out uh, in the colder, dark.
yeah,
You've also got the nematodes, who then can be on board on an autonomous system. The autonomous system will work on a geolocation of moving around,
yeah,
finding the slugs, and then there and then it can spray the nematodes in that area of the slug's location. With that, the slugs being in that location, they don't move far. The slugs themselves, they stay pretty in their own home
yeah.
area. Um, yeah, we spoke about Professor Keith Walter's work as well.
Well, yeah, and
And that's working with us.
yeah, of course, and that's a really important
Yeah.
part of the project. So the other element, as I mentioned earlier on, briefly, that we're looking at patch location and
Mm,
looking more into slug behaviour, how they behave. And this is so Keith Walters at Harper Adams University. He's done decades of work, I think, in this
mm,
field, and established that slugs gather in patches.
mm.
And and you can actually relate to where they gather in the field to specific soil factors, and that is the other element of the project that we're developing. We're doing some farmer trials with 30 slug sloughs across the UK,
Yeah.
looking to. We know that they gather in patches. We have some really good information about that. The point of the trials is to gain a better understanding of how those slug patches form, where the edges are. Where can you
Mm,
reliably turn the slug pelleter on and off in order to treat the majority of slugs that you have in your field. And
yeah.
and I think the results, well, not the results necessarily, but the evidence that Keith has built up over the years, plus the initial trials that we've already done, will indicate that we can really clamp down
Yeah.
on the amount of pellets that we
And that
apply
work
with
is
this.
incredibly complementary to the work where we're working synergistically with
Exactly.
together with
Yeah.
the autonomous system with the nematodes, because we can overload that knowledge and skills and that knowledge and work on different areas, and it just, well, it brings so many different costs down once you've, yeah, I suppose knowledge is power in that respect.
Yeah, so I mean, I think we're talking here. The ultimate vision, I guess, is that rather than going out and willy nilly spreading slug pellets everywhere, you have an autonomous bot that sits there, and we're talking about creatures here that come out at night,
Yeah.
basically, aren't we? So you have an autonomous bot that sits in your field, and it goes out at night, and it knows where the patches are in the field. And
Yeah.
it goes to those patches. It doesn't have to visit the whole field; just goes
Mm,
to those patches, finds a slug, and spot applies it with these nematodes. That's
mm.
am I right? That's where we're the
Well,
ultimate aim, isn't
it
it?
depends on where we're at along the way, and also how crazy the weather is and how high the slug populations are. Initially, as you go down the line with
Hmm.
the autonomous system, we'd probably check the entire fields and everything first, and then work with the data, Keith, because we want to double check and everything. But yes,
Yeah,
that is the idea in the end. As a commercial service, we can narrow these things down and have it spraying and only spraying in those areas where the higher locations of the slugs are and things.
yeah.
Mm.
Well, so we're a
No
year into
waste
the
of nematodes, and we
yeah.
get the slugs. Sorry, yeah.
We're a year into the project. We're just over a year into the project now,
Yeah.
Kerry. So where are we at? What have we actually achieved so
Oh,
far? Particularly in this area, and what challenges have there been?
okay. Well, we're really lucky in this project because it's such a big project. As you said, there's 7 different partners,
Hmm.
and we've got your farmer network. So we've got all these experts that are freely available to us, and we're utilizing them left, right, and center.
Hmm.
So we've got John Ennis who's doing the slug resistant wheat. We've got Keith who's working on the patch location. We've got UK AgriTech who's working with the phenotyping lab, finding that actual location of the slugs and detecting slugs as well as slug damage. So we've done quite a bit of work in the labs, and we've actually trained a model that recognizes what wheat stubble is, what rock is, what slugs are, and we're feeding this now into the next work that we're going to be using out in the farms.
Right.
One of the things we did worry about initially, I think there were a few summers back, there were times where it was difficult to find slugs, but it was
Hmm.
so dry. Um, I got to admit,
That's not
I've
going to be a
always found,
problem this
yeah,
autumn.
yeah, but I think that's why I was brought on board because I can locate them at night and as you say, they're out at night when no one else is about. But this term we don't have a problem. However, what's kept us going over the last year, an amazing help with the different the slug citizen science and the slug
Oh
sleuths and that
yes,
sending
yeah.
in the slugs. So we've
No,
never been short of slugs.
no, no. Well,
We've just had culture after culture. It's been incredible. So
I'm
if
glad
every
you mentioned that.
experiment has just been ticking along. It's
Yeah,
been
Kerry,
brilliant.
that's a really interesting part of the project, actually. So
Yeah.
you mentioned about the slug-resistant wheat,
So,
and we're working with John Innocenter on that. So
yeah.
this is a type of wheat, Watkins 788. It's an old landrace variety
Yeah.
that is apparently consistently spurned by slugs, according to feeding trials that they've been
Yeah.
doing at John
So
Innocenter. So
So that's
we're
great.
doing
They've had enough slugs. I've had enough slugs, and we've kept our trials going.
yeah, absolutely. So we've and we're actually doing trials. And these are part of the trials that we're doing with our slug sleuths.
Yeah.
Is that they're testing different types. They've got Watkins 788 that they're testing next to their farm standard, and we've also got two what they call rills, recombinant inbred lines. So this is Watkins
Yeah.
crossed with Paragon, which is a more modern wheat, and we've got two lines. One that is even apparently, according to the lab trials again, even more consistently spurned by slugs. This is line 64. And then we've got line 50, which is apparently really, really palatable. We're going to be comparing the slug damage in these different lines. But as you say, to do the work in the lab, to look at because they got
Yeah.
84 of these rills, as they call them, that they've got to
You need a
test.
lot of slugs, and I've needed
Yeah,
hundreds of them for each
yeah,
experiment, sort of thing.
hundreds. We're talking thousands. I tell you,
Yeah.
that's what they need. And so we've, as you say, we've been harnessing the skills and expertise of our wonderful citizen scientists, the Boffin citizen scientists, who have been going out into their fields and gardens and collecting slugs and sending them in for feeding trials. And anyone who would like to join as a slug scout, if you go to slimers.co.uk, then you can join there as a slug scout, and we'll send you out a pack to get
Yeah.
you started. But anyway, I won't
Yeah.
talk
Yeah.
in too much detail about that. But suffice it to say, we've got slugs coming out of our ears. But
Yep.
now the thing is, one of the elements of the trials that you've been, well, one of the elements of the work that you've been doing is, as you say, going to our farms and having a look for slugs. And I have to say, Kerry, this is an aspect of your work that I don't know. Well, you kind of draw us in. This fascination with slugs. We hate slugs as farmers. We hate slugs. But you're coming onto a farm and you're kindling within us a sort of fascination in these slimy pests and
I know
what
I've
their
had a few
behaviour
farmers
is.
out with me in the middle of the night in all elements. I think there was a storm Kathy last year. I had one farmer out with me surviving and flooding and everything with an umbrella and everything, looking for slugs in the middle of the night. But
Yeah. So last autumn, Kerry, you came onto a lot of our farms that our slug sleuths who were involved in the trials were doing, and you had a
yeah,
look for slugs, and you were hunting around at night time, especially
yeah.
for those slugs. And I remember actually when you came to my farm, it was Duroceras invadans or something
Yeah,
was,
that's
was
right.
the,
Durostrus invidens.
yeah, which
Closely related to Durostrus reticulum, normally found in gardens. So a bit odd that you had them all over your farm. We
yeah.
normally find the grey field slug, but you had a lot of chestnut slugs, yeah, running around. I think it must have been something to do with some type of vegetation on the edges or something. Maybe we've got a
Right.
lot of apple trees or something.
I know. When you said Duroceras invadans, I thought, my God, I've been invaded by aliens or something like that.
Sounds a little bit like that.
Yeah. And anyway, I don't know what the population is looking like now, but perhaps we'll find out this
Oh,
autumn. So
I'll have to
just
come and have a look again.
tell us a little bit about what the sort of work that we're planning to do this autumn, well, winter, over this season, should I say?
Okay, well, in the labs, I'm doing more slug damage work. So what I've done is I've looked at slug damage, distinguishing actual whether it be slug damage or the plant's not damaged at all, or whether some mechanical device, because
Mm,
plants respond. Their chemical response in different ways to different
right.
things. And for example, if a plant is chewed on by a caterpillar, it responds and lets off a parasitic wasp will follow and find that the large white caterpillars, and then the caterpillars infect the wasps infect the caterpillars with their eggs and larvae and things, and those caterpillars, that's a biological control of its own, so it
Right.
knocks back the species of the caterpillars. But what we want to do is look at those plant responses under the multispectral bands and see if we can distinguish between pests now. So what I have in the lab is caterpillars at the moment as well as slugs. So it's all gone a bit wild.
Yeah,
So
you're telling me about your caterpillars just before we started. And in fact,
yeah,
just to inform the listeners that as we started, Kerry said, "Do you mind if I just quickly break off? I need to just check on the caterpillars."
they're
And
ferocious.
yeah, well, exactly. So
If they're not being controlled by those wasps, they, yeah. So that's the experiment currently running, where I'm just looking at if we can distinguish between pests, and that is my first trial, sort of
yeah.
with a type of pest. And fingers crossed, it's looking good so far, as long as the caterpillars leave me some leaf to actually image and things.
So can I just? I mean, let's move away from caterpillars and back to slugs. But
Mm.
slug damage. So, are we talking about being able to detect slug damage as
Yeah,
opposed to damage from something else? Is
yeah.
what you're
The,
looking at?
the the reasoning behind that is slugs use molluscicide, or the nematodes, and other insects, or anything other damaging, flea beetle, caterpillars, any
Yeah,
other insects. That's in the insecticides, the pesticides. So if you've got a damage by a certain thing, you don't want to be applying a molluscicide in the area where there's no slugs and it's some other insect or pest, and totally
sure.
different. It's also something that we'd like to look at being sort of a complementary bit on the autonomous bit later on, together with finding and geolocating the slugs precisely where they are and things. It's an element that we're looking into. So that's what we're doing in the lab. So we've got the part where we're further developing the model of actually distinguishing what is a slug,
Yeah.
and we've perfected that in the lab now. And
I see. Well,
yeah,
that's interesting because one of the things that when I've mentioned this project to farmers, they said, "Well, this is all very well, but your research is very dependent on actually finding the slugs in the field,
that's not a problem.
and then applying the nematodes. The slugs got to be there."
Yeah,
Well, I guess the if we can detect the slug damage, we can, in theory, and correct me if I'm wrong here,
yeah,
Kerry, we can, in theory, apply the nematodes to that area in the knowledge that the slug
we
won't be too
could.
far away.
We could, but slug damage so that's one thing I'm going to be looking at later on, is that latency, or how long ago that damage took place, and
Right.
what different responses, because plants actually start sort of fixing
Healing.
themselves up, and yeah, healing as such. So slugs could have damaged and then not be there anymore, or some
Right.
other pest could have come along and damaged. So that may be no good. So if that area was on the out edges of where the slug patch is, but they went a little bit further out because it was nice, moist weather to be out a bit further, and they ventured a bit further. So then you look at that area and you're like, oh, the slug is there, right? Apply the control there. Um, that may be useless again. So it's an
Mm,
overlay of all the different knowledge working together, and then forming a perfect idea of where those slugs are. It's
I
got to work
see.
hand in hand with all of them. So we are investigating and researching all the different elements, getting as much knowledge as possible, so we can hit it with full force with this project.
Yeah.
Because we've got all the partners, we've got all the experts, we've got all the farmers who are experts in their own different things in the farms that I'm learning from all the time at the moment. Bring all that knowledge together, and then we've got quite a formidable force. So we
So,
should have
so,
it then.
so, in terms of how farmers can help with this project, I mean, I guess what we're looking for is lots of images of slugs
Hmm.
and slug damage. Um,
So yeah, so actually, at the next stage is that we're going to go onto the farms. Now, I've been to quite few different farms,
mm,
but I do know things change. For example, you don't put the same crop in the same field and different things. So I know what's
yeah,
on what fields. That was last year and the beginning of this year. But we need to know for the next few growing seasons if they can tell us what's being grown on the different what crops are they going to be growing, what crop was grown last year, or what was the last crop on the different fields. So because, for example, if you've got oilseed rape and then you've got wheat, I think everyone knows there's a little bit more slugs
yeah.
about. Or if there's four different questions that we going to be asking. I think using the husk app to
That's right.
pick
Yeah,
up quite a bit of that information for us.
yeah.
So we're hoping to get all these questions asked, and then choose a number of farms to go do a preliminary data collection from approximately a handful of these farms, and then work wider and wider. But the first questions we're asking is, what soil type do you have? What are you currently growing? what was grown before and what type of tillage, what cultivation. So we know, is it medium, min-till? So we know what kind of stubble's in the background and that, because
Yeah.
yeah, we know what we want to look for, and we're trying to get a nice similar images so that we've got a strong set to work with initially, and then we add to it. And once we've built a very good, a strong training set, that's where we take it to the next
So
steps and go further
when
afield.
you're talking about a training set, what would these are images of slugs
Yes,
in the field, basically,
yes.
what we're talking
So
about.
we want a training set. So we're going to be trying to go for the first lot of training set. We're going to try one crop type. And so we're not going to go loads of crops, and
Mm,
the images will be too different. But for our first proof of concept, we want one crop type. We want one type of cultivation mode.
mm,
We want, if what's previously grown, we want kind of the same things in the background and that.
yeah,
What we want is within those types, there's going to be lots of variation anyway.
yeah.
So we're going to keep some variables the same, and then we got other things that we're going to let go wild, so we can build a strong thing on those variables. Does it,
I see.
I
So
hope
we're
I'm
talking,
clear.
we're talking about lots of taking photographs in the field, basically, of slugs, I
Yeah,
suppose. So, and is that going to be? Are you going to be doing that work? Are we going to be getting the farmers to do that work?
it's going to be a combination. Hopefully, I'm going to get some people out there again.
Great.
So, what we're hoping to do is get a few rigs to get the actual data collection set up, and they're going to be simple things that we'll be taking out onto the different farms.
Right.
It will be at night, and
Yeah.
it might be quite like a disco party with the different flashing lights, because we have to use red, blue, green.
Right.
So, it will be interesting,
Yeah.
and yes, we are going to ask the farmers to help us with some of it as we go along. But you won't be standing there all night long trying to take pictures of slugs on the ground.
No, well, I mean, this it'll be a fascinating
Yeah,
part of the trials, and of course, the thing about the boffin trials is that farmers are rewarded for their work, and we have, as I say, 30 slug sleuths who are paid to do on-farm trials, and so this will be another trial that they'll be getting involved in, and there will be payment for these farmers.
well,
But listen,
that'll be great.
this is just about all we've got time for in this episode,
All right.
Kerry. But before we sign off, I just wanted to ask you, where are we heading with this project? What do you think? What impact do you think that this project is going to have? Do you think?
Well, I suppose we're going to think about the number one pest in the UK at the moment is
Mm,
slugs.
yeah,
Is one of the biggest pests, and pretty strong in quite a few other places in the world. And when we think about this weather, it's slug Armageddon at the moment.
yeah,
So this project couldn't come sooner.
yeah,
That is the one thing about it. I know there's a constant struggle of people having to produce more. Farmers are having to produce more right now with using less chemicals and with using less resources. So it's a constant battle of things right now. So the fact that we could find something that's not detrimental to the environment, the impacts of it is good. The costs, we if we narrow those, lower those costs down, because the rising costs are going up anyway.
yeah.
You produce more actual product, the costs going down, it will it'll be sustainable and cost effective.
Well,
So we
I have
couldn't,
to
yeah,
say, yeah, I mean, the thing that really excites me about this project, Kerry, in
yeah,
particular this area, is that this is a totally new form of control for slugs
yeah,
that we're introducing here, these nematodes. Well, it's a new form of control for slugs in the arable field.
yeah.
They've been used for decades, I think, in gardens and so on.
Over 30 years.
Yeah, and
I think they started looking at producing about 30 years when they started first investigating.
really,
I
well,
did a
there we are,
rather long
and
review.
also in horticulture as well. I think in certain horticultural systems
Yes,
that they're used, but
but
not really in our because it's so prohibitively expensive. But we're developing the systems they're going to be using, and that's what's really exciting here. It's a totally new system, and it could be, really revolutionary in its control. In terms of once we've understood a bit more about how nematodes live in the soil and live with slugs, and understood a bit more
yeah,
about slug behavior
yeah.
as well that we're doing as part of this project. And
And
the
it's
other,
fundamental to the existence of farmers
yeah,
to get those things right.
well, exactly, and
Yeah,
that's the exciting thing about this project for me is that we are at the end of the project, we're going to have some services that will be available for farmers that they'll actually be able to roll out onto
yeah.
farm. said he confidently.
No, it'll be economically more viable.
Yeah.
It'll be environmentally and societal. The benefits behind it will just be brilliant.
Yeah.
So, the one thing about working with the UK ATC is that I get to see the beginnings and of the formings of really incredible ideas, because we do a lot of these kind of
Mm.
projects, and I hear about them going along, and then I see people bragging about what they've done, and I can't wait for this project to the point where I'm like, oh, big brag.
Yeah.
I can do it.
Well,
I've already done a few internal conferences and things, and we're getting all very excited by this one.
well, this is where our objectives align. I can say, Kerry,
Yeah,
because and that's exactly what us at Boffin Farmers, we see these fascinating new innovations coming out, and we want them on our farm. We want to help shape them,
yeah,
and UK ATC, as you say, are absolutely at the forefront of that, and we're
yeah,
involved in quite a few projects with you. And
yeah,
the great thing is that you get to see these things first, and you can
yeah.
introduce them to us, and then our farmers can get to play with them in the field. That's
Yeah,
what
have
we like
a
doing.
slug autonomous, fully monitoring and precision application system. It's going to be amazing.
That's right.
Slug robot.
That's what we, I think that's the aim, isn't it? That
Yeah,
within a year, hopefully, as said he,
yeah,
crossed fingers and all sorts of touching wood. Within a year, we should have something that's actually working in the field,
yeah. We
and
definitely
that's
will.
going to be such an exciting prospect. But anyway,
Yeah, we're bouncing
that's
ahead.
all we've
Yeah,
got time for from the Boffin Buzz this episode. Thank you so much, Kerry, for joining us, for
great.
bringing your expertise
Oh, thank you for
about
having me, Tom.
nematodes, about slugs, and so on, and your experience in the Slimers project.
Lots of fun to have a talk. Thanks.
No, not at all. Well, thank you very much, everyone, for listening.
Oh,
And don't forget that Basis members can now claim 1 CPD point after listening to both episodes. So there was episode 1, that was the other episode we had. This is episode 2. And at the end of each of those episodes, there is a CPD code, and if you join the two together, you can claim your basis points. So the second half of the code is, grab a pen and paper and get ready for it. The second half of the code is /2425 /K for kilo. So that's /2425 /K. Now you take both of those two parts of the code, join them together, email it to CPD, that's Charlie Papa Delta, at basis-reg.co.uk. So that's CPD at basis-reg.co.uk. And just to remind you, that hyphen in the middle there is the small hyphen, not the underline. So that's CPD at basis-reg.co.uk. Join the two bits of the CPD code together to claim your point. So thank you very much to Kerry. Thank you very much, everyone, to listening. And join us again on the Boffin Buzz.
Cheers.